So, all right, so let me just take another 10 to 15 minutes to explain the molecular basis of long-term adaptation. So this is what I was telling you in the first half. If you take a wild-type strain, it grows almost within a few minutes, responds to galactose within a few minutes. If you take a Gal3 strain, which is a mutant, it has long lag, in terms of days, in fact. That's because of the presence of the redundant Gal1 signaling protein. If you delete both, the cells cannot be induced at all, okay? The question then is that, why is that a Gal3 deletion strain, despite having a redundant signal transducer, why does it take a long time? So this problem was sort of, people tried to understand this problem in early 50s, 60s, 70s, and they came up with different explanation, okay? The explanation was the following. So this is the, just imagine this, each circle represents a cell. So this is the wild type strain. The moment you add galactose, Gal3 senses that and all the cells are turned on. This is a wild type strain. What was proposed by this person in 1979 to explain this long-term adaptation is the following. What he assumed was that in a Gal3 deletion strain, when you add galactose, in each strain, this Gal1 builds up very slowly over a period of time, and that keeps increasing such that moment the Gal1 reaches a threshold, it senses galactose and the system is on, okay? This was the explanation given by Jim Broche in 1979. So this is sort of a graded concept where all cells respond in a similar fashion, okay? But there was no experimental evidence to this idea, and 2005 we came up with a totally different explanation to explain this long-term adaptation phenotype. We said this is not how the system works. The way system works is that in a Gal3 deletion strain, out of this many cells, in one cell, the amount of Gal1 present is above a threshold to respond to galactose. That cell keeps multiplying, and eventually these cells take over these cells. So this is a totally different sort of an explanation, and we published this in Phelps letters. There was no experimental evidence in this paper. We came up with a theoretical argument. But then question is, how would you detect this cell, which is supposed to be present in very small fraction? How would you detect using techniques that are not available to us? So it took...
but the results of this should depend on the growth curve.
That fits with the theory. Oh, that we have no problem with that. I mean, that's what we explained here. So the question is to provide experimental determination. In fact, when we tried to publish this, the reviewer said this is only a theoretical paper, we cannot publish. Then we fought back and got it published because the theory appeared to explain the phenomena quite nicely. Then it took almost 3-4 years for us to come up with an experimental system to detect this. It turned out that experimental system that we devised was so simple, you didn't require any complicated experiment. Simply by counting cells you can demonstrate this, okay? So I'll just show you what we did. So this is the phenotype. This is the phenotype what we demonstrate on the plate. So the question is now, how do you determine, or how do you provide support for the second hypothesis? What you do is, you take the wild type cell, pre-growth in glycerol, take Gal3 cell, pre-growth in glycerol. Glycerol is a neutral medium. The cells are normal. Now, what you do is, you take X number of cells, there is 1,000 cells from here, you plate it onto glycerol plate. So I know how many cells I have plated, or how many cells this aliquot has by counting the number of cells here. I plate the same cells here, which means if there are 1,000 cells, I should get 1,000 cells which grow on galactose. That means all 1,000 cells respond to galactose in a wild type strain because you have sufficient amount of Gal3. Every cell responds to galactose. Now, if what we are saying is true, there's only small fraction of cells that respond to galactose by virtue of the fact that we have, in that small fraction, there is sufficient Gal1. If that is true, if I plate 1,000 cells here, I should not get 1,000 cells here. I should get, let's say, 5, 10, 15, whatever number, depending upon what that initial number is. If, on the other hand, if what Jim Broche says is correct, then if I do this experiment, if there are 1,000 cells here, I should get 1,000 cells, but the growth of 1,000 cells will take very long period of time. Whereas according to our model, the number of cells that would grow are a small fraction of the total numbers of cells plated, but they would grow very fast. When we did such an experiment, it turned out that in a wild type strain, if I put 1,000 cells here, I get 1,000 cells, clearly, no problem. If I take these cells, put 1,000 cells, put the same number of cells on galactose, I get only 3 colonies. I get 1,000 colonies here. So our explanation is that the 3 colonies that grow here have sufficient amount of Gal1 that is required to sense galactose, and those cells keep multiplying and eventually they populate, and that's why you're getting long-term adaptation phenotype. In other words, if I inoculate 1,000 cells of wild type, all 1,000 cells will respond immediately. Whereas if I inoculate 1,000 cells of Gal3, I'm actually inoculating 3 cells. So 3 cells will become 6, 6 will become 12, 24. So that takes long period of time. That is the reason why we get long-term adaptation based on this experimental evidence. But this is a phenomenological evidence. We don't have.
But can't you measure this by looking at the
No, this is an experiment.
Yeah, but why don't you follow the distribution of amounts of, look at cells, population of cells, and how many cells have X amount of GAL?
No, but the amount of Gal1 in these 3 cells is so small,
yeah.
You cannot determine that experimentally. Because you require small amount of Gal1 to turn it on.
I see.
The moment it senses galactose, the Gal1 through autoregulator mechanism goes up. But 3 cells present in this medium have very small amount of Gal1. I do not know a priori which cell has Gal1. I am trying to deduce that using this experiment.
And you may have answered his point. These are clones right now. You're subcloning them, aren't you? When you dilute them now. So when you say 3 cells, do you mean 3 cells in a given colony?
No.
3 separate. So he is actually looking at single cells.
Yeah. Oh, yeah, right. So I call this as poor man's facts analysis. A single cell. This is single cell analysis.
And if you were to take cells from these 3 colonies and culture them,
I'll do that. Yeah. So now this is only a phenomenological experiment that sort of. The inference is that those 3 cells have sufficient amount of Gal1 to turn it on. Now, we did a facts analysis where you hook on GFP to this wild type and GFP to a Gal3 cell and integrate it to a separate locus. You do a growth curve of this and this. This is the pattern. What you do is, I take cells at this time point and this time point. The idea is that if what we are saying is true, this population should have fully induced cells as well as cells that are not induced at all. That is the expectation. So this is the wild type picture where if you take this many cells, every cell is induced. Here, there are no non-induced cells. If I do a similar experiment with respect to this, you can see here out of these cells, only a small fraction of cells are induced and all other cells are not induced. We have done this experiment by taking time points at different time points, and you can see as a function of time, these cells keep increasing, which shows you that you have 2 populations, one which is fully induced, one which is not fully induced. So this is sort of a binary response. So this is the answer for the long-term adaptation phenotype. So essentially, now let me address your question. So you have in that population 2 types of cells. One is induced, one is non-induced. So what I can do is I can fractionate these cells and these cells. These are fully induced. I can do sorting using facts analysis. So I separate these cells from this. I make sure that they're separated. I take this population, again grow it in many number of generation in glycerol. Okay, so they are now going back to their original self. I again now induce. When I again induce, I get the same population. Probably this answers probably your question that you were trying to say.
This question.
No. In the previous session, you asked a question.
The Novi corner. It's exactly the same.
Yeah, exactly. So you can take this induced cell, you can completely de-induce it. Again, it will give you 3 out of 1,000. So that's how it works. Okay. So that is the explanation for long-term adaptation phenotype. Yeah.
so he said it's all
all or need no intuition. But there's one cell which has a light green.
Yes, good point. So by and large, yes, all or none, but there can be some cells which are probably not as much induced as fully induced.
But I mean, that's a.
So when you do a facts, no, it's not error. When you do a facts analysis, for example, here, this is your distribution. This is your intensity.
Yeah.
So the intensity of these cells and this intensity of these cells is different. But
There's a clear bimodal.
heterogeneity. In fact, when you overexpress or in constitutively induce galacty instead of through the autoregulatory mechanism, the width of this curve increases. That's why you see the heterogeneity increases. So galacty autoregulated loop is required to reduce the width of that curve. Okay.
Not really a bimodal. Maybe there's some bigger peak.
Here.
Yeah, the bigger one. Yeah, or even the second where the cross is. Yeah.
There's a small.
Small.
Yeah. This
So
is.
is there some significance to that, or?
I mean, if you want to give some significance, and you can probably try to figure out what's going on there. This we keep observing. It could be a genetic. Suppose I do this experiment in different laboratory strains, you may get a different pattern.
Can you repeat this thing? I didn't get it. I didn't follow.
Okay, now. So let me go back. So if I take this population here, I get two population of cells here, right? This is not induced, this is induced. Now what I do in the next experiment, I separate this guy out. This is fully induced. I put it into the medium where there is no galactose. It again goes back to the normal state. All the gal genes are off. Now if I again add galactose to this, it bifurcates into non-induced and induced. So it can go through the cycles. This phenomena is reversible. Now, since that you're asking the question, the point is, if I now shift it to glycerol medium, how many generations should I grow it to make sure that it goes back to itself? This is called as cellular memory. So people are working on that sort of aspects. Cellular, how does the cellular memory get established and all those things, but I will not touch upon that. But anyway, that's the point. So the inference is that isogenic Gal3 deletion cells exist in two distinct states. This is due to the stochastic expression of Gal1 above a threshold only in 0.3% of the cells. That 0.3, it's fixed. Anytime you can, repeat this experiment, and it turned out that this experiment was repeated by some other group for a different reason. I will show that result. So this is the pictorial sort of representation of this phenomena. This is the cell identity. Each dash here represents each cell. So these cells will have different Gal1 expression in the absence of galactose, right? Only those cells which have above a threshold, they will see galactose, they will sense it, and they will turn on the switch. So this is the explanation. Now the point is that this threshold can be increased or decreased by different mechanisms. Now I'm coming back to the original Monod's observation where he said this idea of changing the activity function of a gene by changing the concentration or by allostatic regulation. Okay. The next paper which I am going to show you is that he showed that by knocking down a non-coding RNA which traverses across the Gal1, he can increase this number from 0.3 to very high number by changing the expression. This paper appeared in 2015. What he did was, this is the Gal1 and this is the Gal1 transcription. This is the intergenic promoter. People had identified a non-coding RNA which starts from here, goes all the way up to Gal1. The function of this non-coding RNA was not known. Okay? What these people did was, they did an experiment where they knocked down this non-coding RNA by some mechanism, and now monitored what happens to the cell phenotype under two conditions, under induced condition and under induced condition in a wild type background, under induced condition in a Gal3 deletion strain. Okay? So what you do is you take a wild type strain. This is a control where you don't knock down this. Nothing happens, the growth is fine. This is the wild type strain where you knock down this. There is no difference between these two. That means under inducing conditions, this non-coding RNA has no role. But when you delete GAL3, if you delete this non-coding RNA or get rid of non-coding RNA, now there's a small difference in the growth phenotype here. Now what they did was, they did the same experiment assay that we developed, and they counted the rapid inducers. In a GAL3 strain where this non-coding RNA is not destroyed, in a GAL3 strain where this non-coding RNA is destroyed. The moment you destroy this non-coding RNA in a GAL3 deletion strain, the number of rapid inducer goes up from 5 to 35. Meaning that the function of non-coding RNA in the uninduced condition to make sure the basal expression of GAL1 is kept as low as possible. The moment non-coding RNA is destroyed, the basal expression goes up, but that can be only detected in the GAL3 deletion background. So now we have many explanations as to how the expression of gene gets regulated. Okay, there are many mechanisms that are required to keep the switch on or off. So this is the explanation for the expression change and functional difference, the first concept which Monod developed. Okay, now the second concept where you don't change the concentration of the protein, but you change the activity, right? So I was telling you that GAL1 is a bifunctional protein. One of the activity of this protein is to catalyze the reaction where galactose gets phosphorylated to galactose 1-phosphate in presence of ATP. The other function is totally different from a catalytic function where it binds galactose and ATP. Same substrate it binds, but it doesn't catalyze the reaction, but it binds the GAL-AT downstream repressor. So same protein performs two functions. Intuitively one can think that it's possible that if you take a population of GAL1 molecules, 50% of the molecule do transduction, 50% of the molecule do catalytic activity. That's one way of thinking. The other possibility is that each protein molecule goes through dynamics changes. Once it functions as an enzyme, another time it functions as a signal transducer. Now how do you address this issue? It's very difficult. There is literature, not in a single paper is there, which addresses the basis of how a protein functions or performs two different functions within the same protein sequence. There's no information in literature so far. So we thought this is a good model to sort of address this issue. So this is sort of a snapshot of the structure of part of the structure of GAL1. This is ATP. This is galactose. This is one of the residues I have highlighted for this talk. Now you can see this is terminal phosphate. This is the hydroxyl of galactose. The distance between this is 7.2. Between the terminal phosphate and the hydroxyl, anomeric hydroxyl of galactose. This is where the phosphate gets transferred to convert galactose into galactose 1-phosphate. Now it's known that this distance has to be within certain limits in order for the catalysis to occur. Less than, let's say, 4 to 5 angstroms, the catalysis can still occur. But if the distance becomes more, it cannot catalyze. Okay? So what we argued was, GAL1 goes through dynamic conformational changes. During these conformational changes, the distance keeps going back and forth. Right? So if it becomes more than 7.2, it functions as a signal transducer. If it becomes less than, let's say, 5 or 6, it functions as a catalytic molecule. The question is, how do you sort of establish this observation or idea? So one of the ways of doing this, yeah. Between this? You mean you're talking about this distance?
Yeah. So
no, I'll tell you, this is just a snapshot.
Okay.
Okay. So the question is now, how would you establish whether this protein, if you subject it to a molecular dynamic simulation, you get different conformations, right? So then we can sort of probe whether does it go through that changes in the protein structure such that at some time event, that distance is more, at some other time point, the distance is less. I can do that. I can take a simple wild type GAL3 protein, GAL1 protein, which is bifunctional, subject it to a MD simulation study. This is what you observe here. This is a normal wild type Gal1. So what we plot here is this distance as a function of time, and the protein goes through motions, conformation changes. Whenever it goes above this distance, it cannot catalyze. So units of here and angstroms. Yeah. So this is time. So what our assumption is that when the protein exists in this conformation, it cannot catalyze because the distance between ATP and galactose is more, so it must most likely be transducing the signal. So that's okay. But it's very difficult. I mean, you are talking about measuring the distance between this phosphate and this hydroxyl. I do not know how one can do FRET for. Yeah. So, the problem with that is you have to put probes, and then you do not know what happens to the protein. All sorts of complications. Yeah.
So, can the protein be crystallized in these two different states? And then
I am not a great sort of. I went and talked to crystallographers on this, and they said you crystallize this protein and that protein. I didn't believe too much on that because it provides you only a static structure. We have a crystal structure of Gal1. Static structure doesn't tell you anything unless you are able to sort of establish different crystal structures, but you do not know what sort of structures you get.
So, forgive a more basic question. So, you claim that this is a bifunctional protein.
That is known.
That is known. So,
beyond doubt.
Okay. So, my question is, usually the relation between the structure and the function of a protein, is this mapping usually 1-to-1, or do we know?
Yeah, monofunctional proteins. There are many. Okay, let me tell you this. Suppose if I give you a protein, you know a function of the protein. Okay, you can crystallize it, you can study structure and all, but you do not know what sort of a second function it has. There is no a textbook protocol to detect the second function of it. Most of the time, the bifunctionality is detected by serendipitous observations. In this case, it so happened by genetic studies we came to know Gal1 is a bifunctional protein, or else there is no way of detecting other than catalytic activity that it has second function. Of late, the database of moonlighting, they're called as moonlighting proteins. The database of moonlighting proteins has gone up, but still there is no clear understanding as to how, given one protein, how does it functions two reactions or functions which are totally independent.
So theoretically, it is also possible that it's
you can
more
have
than
even
two functions.
three, four activities.
Okay.
It's theoretically possible, yeah. So the point is that we did the molecular dynamics simulation, but this doesn't give us too much of confidence. So what we did was then, we should try to isolate a mutation in Gal1 that increases only signal transduction activity. Okay? If I isolate that mutant, if I subject that mutant to such an analysis, I should observe more often that protein should exist in this part of the figure, if that is true. But the question is then, how do you identify a mutation in Gal1 that specifically increases the signal transduction activity? There's no way of identifying such residues where you can go and mutate and increase only one activity without losing the other activity. So we did a simple manual sort of bioinformatics analysis to identify what residue that we can mutate in order to increase the activity of signal transduction. So we have 3 galactokinases. One is Saccharomyces cerevisiae, one is K-lactose, both are bifunctional, and we have Gal3, which is monofunctional. Okay? You just align them because the number of amino acid residues is by and large same. Look at residues which are present both in Saccharomyces cerevisiae and K-lactose Gal1 because both are bifunctional. And look at whether that residue is conserved here.
Which is the last line?
This is Gal3 of Saccharomyces cerevisiae, which has no kinase activity. This has only signaling activity. Whereas both these are bifunctional. So our job is to identify a residue in Saccharomyces Gal1. Upon mutation, it should give us more signaling function. Okay? So you just manually look at the residues here. Turns out that these are almost 70% identical sequence, 90% similarity. So we have a residue which is identical here, but here it's different. If that is the case, since this doesn't have kinase, this has more signaling activity, what will happen if I replace this residue here? Will it give more signaling activity? That was the logic we used. So we also did this at a larger scale.
I'm still unclear what the three lines are. First line is Saccharomyces, then Saccharomyces, and the last line?
This is the Gal3, which is a paralog of Saccharomyces Gal1.
Taken from where?
Saccharomyces. Okay. So we tried to do this, and using this approach, we identified one residue in Gal1, that is this tyrosine 441. We replaced this tyrosine 441 to alanine with the hope that that mutant version of Gal1 will do a better signal transduction activity. Okay. And before that, we do a molecular dynamic simulation of the mutant. So this is the wild type, this is the mutant version. Now clearly you can see the mutant version more often exists in this state, which is a non-catalytic state, because the distance between ATP and galactose is much above 6. Now, can I now determine the rapid inducer numbers in a strain having this as compared to this? What I expect is that if this is true, I should have more than 3 out of 1,000 rapid inducers here.
Is Gal1 or Gal3?
No, this is Gal1. So the point I'm saying is Gal1 is a bifunctional protein, right?
What have you done to Gal1?
I have mutated.
What you said was Gal3. You identified from Gal3. What is the difference?
True.
And then what you've done is
I take this residue and replace it, or I identify that residue and change that residue. Essentially, that's what I'm doing. So,
Understand this. So am I supposed to compare the two graphs and say that the lower graph is the system is spending more time in the other state?
yeah, right, exactly.
The time scales are, this is like 650 nanoseconds.
Yeah, this is 1 microsecond, but we have done this for a longer period of time. I have not shown that data here, but by and large, it does show that the conformation exists in this state more often as compared to the wild type.
Okay.
But in fact, these days people say, I'm not a structural biologist, but people say don't do it for too long period of time, because if there's an error right in the beginning, it gets carried over. Yeah. So I'm totally new to this area, but that's the issue, right? Instead of doing this, you do many repetitions for a shorter period of time. That's what people advise. Yeah.
Just one point regarding the direct comparison of the same frame between two graphs, upper and lower. This cannot be compared
You cannot
because
compare the same frame.
yes, I think that's what
Is that what
he was
you're saying?
asking.
No, same frame you cannot compare, because your initial structure itself could be different because of this mutation.
Because
You cannot compare this conformation to this. No.
But I think the point comes through clearly, that the amount of time.
Absolutely. Well, there's
Yes.
there's no question about that. Yeah. There's no question about that. No. I'm sorry. This is, I can represent this. No, this x-axis I can represent either in frames or in terms of seconds or time.
fraction of time.
Exactly.
So you can cut that up by 4, right? You know, there's a
So I'm not.
First of all,
the number of, there are more than, 2 states that are being visited, okay, in the bottom curve. It's spending time at the distance. There's so many different distances, right? So, I've done MD simulations, and I would not say that it's just a 2-state problem.
No, what
Okay,
is
that
the
is-
number of states? Just the literal statement, the fraction of time spent above the line per unit time is higher in the lower graph than the upper one. Isn't that correct?
Okay, so first of all, I don't know how you can get 1 microsecond of data from the above MD. It's an atomic,
No. So we
atomistic
continue
molecular
the
dynamic
simulation
simulation.
for 1 microsecond.
Usually,
So
okay.
we had done
Okay.
MD simulation for the GAL3 is
Yeah.
different. I did not discuss
Yeah.
that. So
Are these coarse-grained simulations or are they atomistic simulations, all atom simulations?
all atom simulations. Yeah.
Yeah.
But okay, I mean, some of the reviewers say that look, the time duration what you have done is for very shorter duration. You cannot.
Yes, that's a concern. That's why MD
This data
simulations
is not
are
published
good.
yet.
Yeah.
This is yet to be published, but this point I will consider.
I would agree with them.
Yeah.
How big is this protein which is
0.29 amino acids. Yeah.
and along with water?
Yeah. You equilibrate in the beginning and then subject it to. But the point I'm saying is that this, what is plotted here is the distance between ATP and hydroxyl. Okay. So we are simply fractionating the conformation to two states. So now the important point is, this is as far as the MD simulation data is concerned. Now, if you do a rapid inducer assay, the assay that we did initially, where you do this for the wild type as well as for the mutant, when you do that, you clearly get the number of rapid inducers in this mutant is much more than this. Here you get 20, here you get 3, 3.5, whatever. We have repeated this assays number of times, and this number is quite robust. So by introducing this mutation, you have increased the signal transduction activity, and we are yet to go and check what happens to the galactokinase activity. Is it at the sacrifice of galactokinase activity that the signal transduction activity goes up, or the galactokinase activity is still retained without disturbing that activity, this function has gone. So at least we have some idea now how a bifunctional protein, especially in this sort of situation, functions in two, or carries out two independent functions. This is fluctuation test. All right, so I'll stop here. And before I take any questions, let me just thank all the students who have carried out this work, and the financial support is from DST, CSR, and BRNS. I shall stop here and take questions.
Like a clarification from Madan, is the point
So,
molecular dynamics for a microsecond? Is that the point?
no, because if they're small enough, it depends on what you mean by molecular. If it's an all atom simulation, it takes longer. If you do a coarse-grained simulation, it takes, you can, I mean, if you do an all atom simulation, you can't run it for too long. And if you do, because the unit of time is very tiny, is very small. But if you do a coarse-grained simulation, then you expand the unit of time and you can.
No, you cannot do for long period of time, meaning that it's not valid, the results you get from that, or?
You can't reach that unit of time. Because the unit of time is a picosecond. So you can, so 100 frames in units of picosecond is something, whereas the unit of time in the coarse-grained simulation could be, depending on the coarse-grained thing, could be a microsecond or a tenth of a microsecond.
For estimating a distance like this, what kind of simulation do you need?
So I mean, for this, I think you'd need an all-atom simulation because you're asking for the details of a chemistry. So the method of this thing is not a problem. But you're comparing a 1 microsecond to. I just wanted to, you said that I can compare the fractions of thing. So imagine this: you're doing a coin toss, and you're doing million times this thing, and you get 50% heads, 50% tails. Then I look at another person who just does it 10 times. Okay, and all 10 could be heads. Now, there's no sense in comparing one and the other.
Yeah, that point is valid, but the result here is exactly in the other direction.
Not done, no. This is just one run.
It's only one run, but it's how many runs is it? Right? So
Yeah, this is the data what's obtained. I mean, I'm not an expert in this simulation. This was done in
We've done
CDAC.
these things, yeah. So it's
Sir, can I ask?
and even if you do it, you can calculate how many times you need to, if you want to compare the data from 10 coin tosses to a million
No,
coin
one of the-
tosses. What it would be. This is a single run, of course. Single run.
Even from the graph, you can see, like Madan was saying, it's the other one. It's probably not, the black line is just something that we have drawn. But if you really look at the fraction of time spent in the distances, there's definitely more than 2 states.
Yeah, that could be. No. I agree with that point. So, for example, no. That's fine. So, for example, this conformation is different from this.
Even about.
There's no question about that. But what I'm saying is the fraction of time the protein spends in this conformation is much more than the fraction of time this conformation is. graph. True. Yeah. If you just draw these two lines here.
Yeah.
But what I'm
Statistically significant if you want difference.
Sir, have you, you had mentioned that you have also done MD for GAL3, so that,
that's a separate study. I can discuss about that later on.
okay. So it's, okay.
There the question
because
was totally-
ideally,
there the question was totally different.
okay,
Whether we were trying to establish the validity of conformational shift mechanism
yes,
in terms of trying to explain the allosteric regulation.
okay. So also the MD for such a 500 amino acid long protein can be possible for 1 microsecond, just that it takes long time. And also the snapshot can be
No, that's what I understand.
So it is
I mean,
possible to
probably I
do
think
MD for such a long period. The main constraint is basically the snapshot input what we are giving. If it is 2 femtosecond, or if you increase to 10 femtosecond, we can still run for long period.
but he doesn't seem to.
And yeah, so
Yeah.
yes.
Any other questions?
Also, okay. I just want to, in the Paradox1, so there are 3 mutations you had mentioned. In one mutation, it is inducible, another is constitutive, and then the third one is non-inducible. So, so is it basically the protein-protein interaction determines that effect? So the first 2 mutations basically affect the interaction between GAL80 and GAL4, whereas the third one, which is non-inducible, is affecting the interface between GAL80 and GAL80.
So this is, let's say, S-1. This is wild type. Okay, this is 4C. Right? So what is your question now?
So the first 2 cases in the lower table,
Okay.
where the inducible and the constitutive.
Okay.
Yeah, so the mutations,
You can go back, yeah.
yes. So, yeah, if I understood correct, in the bottom table, so the GAL3 invariably binds to GAL80 in all the 3 cases. The first 2 scenario, yes, the first 2 scenario, basically it affects the mutation affects the interface region between GAL80 and GAL4. That's why we see this phenotype, whereas in the case of non-inducible, the interaction between GAL80 is GAL80 and GAL80. It's a homodimeric interaction is preserved, so even GAL3 binds to the GAL80, it is not, the inhibition interaction is not removed. Is it the case? That is how I
I did not understand what you're trying to say.
Okay, in that, no sir, it is, it's over there in the picture drawn in the,
Here.
yeah. So basically, if I understood correct, the GAL3 in all the 3 mutations binds to GAL80. Because that's what you had shown in the...
Suppose, for example, if you take this strain, or this strain, or this strain, the
Yes.
only mutations in the GAL3, GAL80, correct?
Yes, sir.
And none of the strains are inducible.
Yes.
What it means is that this binds GAL4, this binds GAL4, this binds GAL4. In none of the cases, GAL3 is able to overcome that repression. That's what it means from this particular data. Is that fine?
Yeah.
Do you
Now,
get that?
yes, sir, I understood.
Okay.
If what I speculating is that if the GAL80 binds to GAL4, and the binding of the GAL80 and GAL4 is basically inhibition, isn't it?
It is inhibition, right?
Yes. If the mutation is making the complex GAL80 and GAL4 is intact. It's like a permanent complex. Then, irrespective of whether Gal3 is binding or not, it invariably binds to Gal4. The Gal80 binds to Gal4. So that, and hence the activation will not happen.
It will not happen. It should not happen here. But what you observe, it does happen.
So so this mutation in Gal4 is basically altering the interaction between Gal4 and Gal80.
Yeah, that is because now
it's a complementary
now
mutation.
Gal3 is able to interact with this. That's how this interaction is disturbed.
Yes.
It's not complementation, it is epistasis.
What she's saying is in the wild type, Gal80 and Gal4 are not tightly interacting. Gal80 can be knocked away by Gal3.
Correct.
Whereas in the mutation, maybe Gal80 and Gal4 are more tightly interacting.
That's the reason why you require more of Gal3 in order to knock down this interaction. That's what our binding experiments suggest.
Okay.
So the amount of Gal3 that's required to knock down its function is the amount of Gal3 required to knock down its function is much more than what is required to knock down this function.
So it's basically to do with the interface region.
Yeah, it's the amount.
Okay. But the last case is basically even though the mutations
here,
are there in both
that
cases.
This Gal3 is not interacting with this at all. That's no problem with this and this. This explanation and this explanation is not an issue. This explanation not an issue. But the problem comes when you have this.
Okay.
That is the crux of the issue. And that's where you get into problems.
Okay.
We can discuss it. Okay, in fact, that is the experiment that we did in vivo that led us to solve this paradox. But the overexpression has to be of Gal3, not from its native promoter, it has to be from an independent promoter so that it's not coupled. That is the mistake the previous people made. They overexpressed from its own promoter, they did not get the induction. That is the issue. Because the amount of Gal3 getting auto-regulated is through the switch. So you have to express Gal3 from a heterologous promoter, so you disentangle that expression of Gal3 from this switch.
So if you take like ADH
now
promoter.
I'll tell you what, I take this strain, it's non-inducible, right? Gal3 cannot bind. What we did was we took this strain, I overexpressed Gal3 over and above this from a plasmid which expresses Gal3 from CYC promoter, and I can make this strain inducible with that. You got my point, no? That is the experiment that gave us clue that this interaction has to occur. Then biochemically they did not observe that. We made sure that it's a concentration dependent phenomenon.
How does a non-coding RNA inhibit?
Non-coding RNA, that's a question which I may not be able to clearly answer, but let me just try. The session at 1:45. Where is that? Let me just see here. Okay, the way it's represented here. See, the lower strand is the coding strand for Gal10. It goes this way, transcription. There's a promoter here on the other strand which drives the transcription of this non-coding RNA. Okay. Now this is the place where you have the divergent promoter. It drives Gal10 RNA. This drives Gal1 RNA. What is sort of anticipated is that since you know this non-coding RNA comes all the way up to here, because this transcription going on, it's not able to turn on this Gal1 expression, which at the low level in the absence of galactose. In the absence of galactose, because of this transcription going this way, there is no basal expression of Gal1. So you knock down this guy, then you can detect that only in this background. You cannot detect this in this background because in a wild type background you have sufficient amount of Gal3, Gal80, Gal2 to overcome this effect. It's a sort of a physical occlusion, sort of, I mean, that mechanism as to how it sort of reduces the basal expression is not clearly explained, but that's what is assumed.
What is the molecular mechanism for that epistatic binding? Do you know?
Conformation.
Huh?
It's protein conformation.
But are the details known of that?
We have the structure for GAL80.
Yeah.
We have the structure for GAL3.
Correct.
We have the structure for GAL3, GAL80.
Yes.
We don't have the structure for GAL4, GAL80, but the structure complex of GAL3, GAL80,
Yeah.
their explanation for that ATS-1 and 3 binding is not right. It got published in 2012. So when we studied the allosteric transition of GAL3 protein,
Right.
we figured out that their explanation is not right.
So,
You understand what I'm saying?
Yeah.
So you have a crystal structure for GAL3, GAL80.
Yeah.
And based on that structure, they deduced the interaction between GAL80S-1 and GAL3.
Okay.
They did not co-crystallize GAL80S-1 and GAL3.
Okay.
They co-crystallized only wild-type GAL3 and GAL80.
I see.
Based on this sort of deduced what could happen if GAL3, ATS-1 and GAL3 interact. And they came up with an explanation. That explanation was not correct.
Right.
I mean, that we know
Yeah.
from our other paper where we
Okay.
figured out how the allosteric transition occurs in the
And so do you know the correct?
Huh?
What is the correct?
Oh, I had to go back. I don't often remember the.
But it's known.
Yeah. It's sort of.
It's just a comment. I wish Mohit was around here because he has worked on this. You know, these remarks that people made about your molecular dynamic simulation may be indicating 3 states rather than 2. You said you have modeled your system.
Just before.
Let me finish my point. This has nothing to do with the molecular dynamics, my question.
Yeah.
You said you have modeled the system. You've
And the molecular dynamics of.
kinetic models, not the molecular dynamics.
I see, okay. The other model, okay.
The kinetic model. Yeah.
Right.
Okay. Is it possible that you have a tri-stable outcome rather than a bistable outcome from your model?
That is the question what he asked in the
From the model, I missed that.
the facts analysis. You may get some different states rather than.
No, this has nothing to do with the molecular dynamics except that someone said.
No, that's a very good question, and that's the question that I would like to pose to the physicists. As far as
graph, there were 2.
yeah. Exactly. So, what I'm trying to tell this audience here is that we are talking about stochasticity in gene expression. Most of the time when you're talking about stochastics in gene expression, you talk about the binding events, low number of molecules, that gives rise to heterogeneity. Nobody is factoring in the noise that gets generated because of protein conformation change. No one has factored in that at all. If you factor in that, probably you can explain many more phenomena in biology which probably have been not explained thus far. I mean, this is my take on this. I mean, it's an interesting type of question that you can address, but the possible states the cell would exist, microstates, can be very high. I'm not.
No, I'm not even thinking of the real cell. What I said was you had a model there, involving.
No, but to answer that question, that different states had to be translated in the model in terms of binding affinities, right?
My question is, just look at the equations that you have as they are,
Okay.
or your geometrical visual picture of the feedbacks involved there.
Okay.
Do they permit 3 stable states to exist?
That might be
Just an analytical treatment of that visual model that you had.
I understand what you're saying, but that will have to go back to the model and see which parameter you will have to sort of tweak in to get that state. But from a protein structure point of view, if there are more than 2-3 states which are active, it gets translated at some time in terms of that affinities, right? Even if 3-4 states are there. Yeah, so one can do that, yeah. Any more questions?
Any more questions?
So I just have little clarification on this. Why do we need to invoke multi-state
multiple states here? I mean, effectively, it's a bistable system, right? You have a whole bunch of things at one bottom end and a whole bunch of things at one top end. Wouldn't that be reasonable to think about it?
No, I wasn't suggesting that you need to evoke a three-state system or a larger number of state system. I just wanted to know what it was likely to be. And if there was a way now, so if you had a three-state system, you could presumably leak from one state to the other more easily than if you had to make a macroscopic switch. This is what I was trying to get at. Mohit, now that you're back, so we are discussing a possible three-state system here.
So I have a separate question related to this though. So you have X number of cells in one state and an X number of Y number of cells in another state. Can you actually change the ratios of that? Because this seems fairly predictable, right? What you're getting.
To the previous paper.
Is it? Okay.
Yeah.
But how do you do that?
By mutation.
By mutate, but you have only two effective states.
We have done that in GAL3. We have done in GAL3. Probably I can show you some. Okay, so now I'm talking about GAL3 protein. Okay, if you take a wild type GAL3 in a cell, it exists in active conformation and inactive conformation to start with when you are growing it in glycerol, right? When you add galactose, it shifts to the active form. Okay, now so either I can convert this state into this state either by adding galactose or by mutating this to this. What you call it? Constitutive mutants. Okay, the concentration of this increases as compared to this. So this is my starting point. This is the whole argument of conformational shift analysis of allosteric, because monomeric proteins are shown to be allosteric. The classical allosteric is MNF model, and no, you have to have multimeric state. But today we know even monomeric proteins show allosteric, and one of the mechanism is conformational shift mechanism. So you have two populations here. If this is true, the corollary also has to be true of conformation state allosteric transition. No one has so far shown whether this is true or not. The reason for that is if I mutate this protein to give me this sort of a conformational fractionation, I'm increasing the inactive protein by mutation. But when you inactivate a protein, people will say it's inactive protein. Where is conformational shift mechanism? That's the question, right? So what we did was, we mutated this into this. We increased this population, but despite that, it responded to galactose, which means that the mutant form, this is the wild type form, the mutant form exists in this state. It's inactive as far as the state is concerned, but it responds to galactose exactly the same way as the wild type. Now let me show you here. And what are the number of mutations that required? You require 3 independent mutations in order to shift from here to here. You put any one of these, it doesn't work. Any or two of that doesn't work. Does that answer your question? We have done the dynamics of this and I have shown why you require 3 mutations and so forth and so on. So this is one of the first examples where you sort of show the both mechanisms, I mean both sides of the conformational shift mechanisms, you know.
Temperatures, external conditions, and
good point, very good point, yeah. So you are saying that this transition is a conformational transition, a temperature dependent phenomena. Yeah, I mean we were thinking of doing that, but then such experiments becomes a little bit messy because then you get an output, then you cannot point out that output is exactly because of this. There can be other things happening, but the point well taken, absolutely, yeah. We were trying to do such experiments, but we didn't really pursue that, yeah.
So in the distribution of 2 states that you were talking about towards the very end, towards the very end, we're talking about 2 different cell populations, the phenotypic heterogeneity aspect.
Okay.
Yes. So in that case, you said that the final distribution of the population comes back to similar to from where you started, irrespective of whether you isolate the green cells or non-green cells.
True. If you start from the starting point, it will have 3 out of 1,000 cells which contain sufficient galactokinase to turn on. You're right.
Right. But when you did the facts and segregated those cells,
correct,
and then looked at the distributions, so was that done at long enough periods of time that distribution is a stable one? Can we call that a steady state, or is it just
yes, we have done it for a long period of time. That's why I was talking about cellular memory. Depends upon, what people do is, not in this context, you grow in galactose, two sets of experiments, then you shift it to glucose, and again you shift it to galactose. The question is now how long the initial galactose memory lasts when it's going through glucose. How many generations it had to. So they sort of use this paradigm to establish the cellular memory. How long this state gets locked, and what are the roles of feedback loops in that process?
Because in many cases where people have looked at these distributions at final as well as initial, so let's say if you have some multi-model population, you segregate those subpopulations, and then see what subpopulation structure you get at the end. Now, in some cases, the population structure is identical to what you started with. In some cases, they're not. So I'm not sure if, when exactly do people typically stop these experiments? Is it when it matches the initial condition, or is it?
That has to be, if you do not have information about the system, you will have to do by trial and error mechanisms. If you have the information about the loops and all, then you can probably rationalize.
Right.
Or else you have to do repeat number of experiments for taking it for a longer period of time. It might so happen that it might get locked so strongly that it may not come back again.
I think the question is that whether it reaches steady state,
yes,
or is this some transient?
Yes, whether this is a transient.
That has to be probably done by trial and error experiments. I'm not sure whether how otherwise you can infer whether it's a...
Because in trying to match this to any sort of modeling approach where we get the steady state distribution.
Yeah, but that will become complicated. What is the type of phenotype you are looking for?
So the second one is often higher tier, right, than that distribution. So maybe
but
that is the signature of the memory. Right?
This is high. No, that depends upon the time, how long you have incubated in a...
Right.
In this case, we incubate for a shorter period of time simply because we wanted to isolate this guy, that's all. Here we have increased for a longer period of time so that we want, essentially I want to demonstrate these two peaks.
It's just intensity, fluorescence. So I just noticed that it's in logarithmic scale, the x-axis. Yeah. So the peaks are really, if you really plot on linear scale, maybe they'll really get stretched out.
Yeah.
No. Also again,
so there is a lot of heterogeneity right here. Normally we plot the mean, and sometimes the plotting mean can be really.
So going back to the differences you were talking about, K lactis and S cerevisiae in terms of GAL3. So when the basal levels increase, the fold change decreased.
Right.
Even
the absolute level also decreases actually, not only the fold change.
Right.
If you remember that graph, not only the fold change occurs, but even the absolute amount of enzyme synthesis also is little less.
So would you say these are causally related? That when the basal levels increase?
That is difficult to answer. It can be just a fallout of, I don't think you can separate, you might be right. I think that's a connection. When the basal level increases, the absolute level increase upon induction can become less. I didn't really think in that direction, but it's possible.
And then the last question, so if you track the number of green cells over time in this particular case.
Okay, as a function of time.
As a function of time, just the total number of green cells. So then if only initially the few cells, the 3 cells which were green and are continuing to divide, then that graph should look quite exponential. But if there are more cells which are switching in the period of time while others are dividing, then that graph will look slightly different.
You're right.
So which is happening here?
No, it's not the other cells. That's contribution coming from non-induced cells switching. I think in my opinion, even if it happens, it's a very small fraction. I'll tell you why I'm saying that. The non-inducing cells sort of, we assume that they don't divide at all because they cannot use galactose as a carbon source. But we are providing amino acids. Cells can utilize even amino acids as a carbon source, but the division rate will be so slow, even if it happens, it can induce. But you don't have to even think about it. You can hold a cell, okay? Imagine, do a sort of thought experiment, hold a cell and measure the activity of GAL1 varying as a function of time. Don't allow it to divide. It may not remain exactly constant, it will go up and down. So at one point it has gone above the threshold. If you provide galactose, it will induce. It's not that...
Just a naive question, will GAL1 also binds to GAL80 or...
Right, true.
It binds?
Yeah.
Okay, if the given, the concentration of GAL3 expression, the protein in the cell is more than GAL1 or
no.
it is equal?
No, it's much less.
Okay.
It's much less under induced condition. Under non-induced condition, GAL1 is very low, GAL3 is high. When induction occurs, this goes little up, whereas GAL1 goes very high.
Okay, so which one exactly, which one exactly binds to GAL80, either GAL3 or GAL1?
There a paper appeared in 2012. What they're trying to say, you induce the system, GAL3 binds GAL80, the system is turned on. As a function of time, the maintenance of the induced state is performed by GAL1, not by GAL3.
Okay.
That makes sense because the induced state, the GAL1 is so much as compared to GAL3, so most often GAL1 binds to GAL80.
So the binding mode of GAL3 with the GAL80 will be almost same as GAL1 with...
Except that GAL1 is not as good a signal transducer as GAL1 in terms of binding.
Okay. Yeah. So is there any structural comparison made?
You can superimpose the structure of GAL1 and GAL3 with RMSD of very, very few,
Okay, very almost, less than 1 angstrom or 1.5.
It's almost superimposable.
Okay, just the constitutively active GAL3, would it perform better with the mellow-bios?
We have not done that. We did not isolate that constitutive GAL3 mutants. Of course, we have not done that, but I'm not sure what is that.
Because if it's constitutively actively, then you have the alpha galactosidase also expressed better.
Correct.
And in a mellow-bios medium, then you have both galactose and glucose both present. So how would the constitutively activity perform in your mellow-bios experiment? Will it be...
But there again, when the galactose and glucose gets produced, the galactose will go and bind the constitutive GAL3.
Yeah, but the glucose repression, does it able to overcome the constitutive activity of this one through GAL3 independent pathway?
Is totally independent of the
GAL3 pathway at all. So the glucose repression does not act through the GAL3 at all. No. So glucose repression will still overcome.
Yeah. That's for sure. Yeah. That's why when you mutate GAL80 and GAL4C, we still don't understand how changing this interaction affects glucose repression. We are not unable to connect this. We try to model the Malibuy's growth curve. So you have the wild type, this is the mutant, this is the wild type. This is in Malibuy's, right? This is the mutant. So I can model this, right? When I model this, I have a value for 84 interaction. If I change this, the only difference between this and this is this interaction parameter. There is no other difference. I try to change this, the model should give us this, but it doesn't give. It doesn't jump from here to here. But in the system we know the only difference between the wild type and the mutant is only this. When I try to plug this in the model, the model is not giving this pattern, which means that this change in the pattern has some effect indirectly on the glucose repression and things like that. So unless you sort of introduce that in the model, you cannot sort of show that. Any other question?
The gene expression level can, in some cases, go up gradually. In some cases, it can go up like a switch, and what controls that kind of thing. And then more generally, regulatory logic and circuits. So this is one example of a circuit that switches on in the absence of a particular. So if you have both glucose and galactose, then the circuit, then the galactose system is basically switched off. If you're depleted of glucose and you have some galactose, then that turns on the galactose system. So it was a kind of complicated, well, not very complicated by biology standards, but that's a regulatory diagram that Bhat showed. So talk about a bit of the theoretical work that's been done on that. And then evolution via gene duplication, that's probably one of the more interesting things to me. And in the last thing about how they feed on melabios, there's some fraction of cells that turn on this gal pathway and produce alpha-galactose, and those guys actually don't benefit because they're doing work for the other cells to benefit. And how do you explain that in evolutionary terms? That's a long-standing thing that many people have worked on. And feel free to stop me at any time. As I said, it's not my work, so I might not be able to answer, but okay. So we'll start with the work that was, I think, Mukund's PhD work. It was from 2001 in PNAS. Okay, so they look at the noise in the expression of proteins, which is what you aim at. And as you know, proteins, it's a two-step process. First, you transcribe to messenger RNA, and then you translate to a protein. But in the simplest case, it is just one gene that might be constitutively expressed. In more complicated cases, which is typical, you have genes that have multiple inputs from upstream, and you have to consider the noise in all of those inputs. But you also have noise in the mRNA expression as well as in the protein expression, mRNA to protein translation. The translation, so the mRNA production is sometimes kind of approximated by a Michaelis-Menten kind of kinetics, where you treat the DNA as a substrate and the polymerase as an enzyme. It doesn't work all that well, but it's kind of a continuous thing. mRNA is being continuously produced at various in various ways, whereas the translation seems to occur in bursts. So the mRNA enters the ribosome and a burst of proteins is produced. So what they find, in fact, is that the fluctuations, so they model this both at a single gene level and at the level of a few simple circuits, and they find that fluctuations are dominated by at the translational noise at the translational level rather than the transcriptional level. But mainly they have a theoretical framework which they expect. So this from 2001, as I said. It's one of the, it's not the first work to do this, but it's one of the earlier works. Along the same lines, okay, this is much more recent work. This is, I think, from 2007 in Cell. This is now moving on to eukaryotes. It's about the Drosophila embryo. And we heard a little bit yesterday about early Drosophila development, and we've heard about morphogens and so on. And the question is, how precisely do these morphogens, gradients, expression levels, concentration profiles need to be established? And this is partly experimental work, and they find that the various noise quantities are all within 10%. I won't go much into this paper, but there are a couple of nice figures from this, which, so this is the usual picture of how bicoid is distributed in the very early cell. It's translated from maternal RNA, which is deposited here and diffuses through, so it's kind of an exponential profile. Whereas hunchback, which turns on a little later, has more or less a step profile. So hunchback is very sensitive to the level of bicoid. If it is above some threshold, you get hunchback, otherwise you don't. And then the question is, how does noise affect this expression, bicoid versus hunchback?
Is the noise intrinsic or extrinsic?
Actually, I can't answer because I haven't read the paper carefully enough. But
both, is it?
Both, I think. Yeah. It was an experimental paper. I think they introduced sources of noise to measure it. But they're really after what's the intrinsic noise in a cell. How does that, in the embryo, how does that affect the further development? But yeah, so Right, this is another piece of work from the early 2000s, 2002, I think. And this is again in E. coli. And basically, they looked at the stochasticity in, they basically engineer, in this paper, they engineer E. coli that can, they're able to detect the noise via reporter genes, and they find that the amplitude of noise is governed by transcription, rate-regulated dynamics, and so on. And they basically say noise is important. It can limit the precision of gene regulation, and it's part of biology. These guys, this is kind of a review article, so they talk about the effects of noise. There's a much more recent article, I think 2010, but they talk about how it can play a beneficial role. So one example was in Let's talk about how some fraction of cells, and not all of them, might be expressing alpha-galactokinase, but that helps the population. So you need cellular heterogeneity even from a clonal population under many circumstances. They also talk about cases where noise actually stabilizes gene expression, which this might be familiar with. So this kind of paper worth reading, probably. Right, so moving on to genetic circuits. This is an oldish paper from Hua's group. I think it's, again, 2002, 2003. What they do is, so we are familiar with various logic. You can think of transcriptional activation as a logic circuit. So, for example, in the case of a single GAL gene, which is controlled by GAL4 and GAL80, you need GAL4 to be there and you need GAL80 not to be there. So that's kind of you can say GAL4 and not a GAL80. You can do more complicated logical functions. And many of these things have been done in E. coli. In fact, I think Mukund was doing something like that in CBS a few years ago. So what they have done is engineer various standard logic gates that are familiar to computer scientists. So A and B means both A and B need to be there to recruit the polymerase. That's the simplest example. And as a function of the dosage of A and B, you get the maximum level when A and B are both high. And whereas you could have a case, so this is a case where A and B kind of need to interact with each other and with the polymerase. Whereas you might have a case where either A or B should be there. This is also a very common situation. They interact independently and either one of them is good enough. So that's the OR function. You can have a NAND function, which is basically, if A and B are both there, they repress. So it is the opposite of this. And similarly, you can have, so this is NAND, this is, and this is basically something like XOR, where you want A and B, A or B, but not both. This is how, so this looks rather complicated, but this is, I don't think this occurs in the real world in cells. I'm not aware of examples. But if you wanted an XOR function, this is what you would do. And there's another way to do XOR. Logically, these two are equivalent, but the implementation is different. So this was an interesting and influential paper at that time. So lots of other groups, yeah, sorry. Sorry, if I'm talking about noise? Yes.
Is required to like remove GAL80 from GAL4 so that you can basically have like galactose coming in and doing its thing.
Right, I've moved on from noise now.
No, I understand that. I'm just saying that, is it possible, as far as we are looking for like problems to solve, right? I mean, the issue is like, can you basically try to like bring in noise effects as far as combinations, as far as these combinations themselves go? And are there some kind of limitations imposed on these, some kind of signaling limitations imposed on what combinations, how do noise effect basically, impose limitations on the kind of combinations involved in a lot of these genetic regulations? That is one of the
yes, questions. In fact, there are, individual circuits have been studied. So in For example, electrical engineering, negative feedback works to control noise, and they found examples of that. So we saw an example of negative feedback where Gal-80, I think, negatively regulates itself. And somebody asks why. It was in the context of a much larger circuit, but if you have a case where you want to keep the expression in some limit, and if the limit, so I think one example is the tryptophan synthase in E. coli. It synthesizes tryptophan when there is no environmental tryptophan, but you don't want too much of it. So then there is a negative feedback where the gene product negatively regulates itself so that it doesn't make too much, but it's also positively regulated by something else. There are other individual examples. I am not aware of any general principles you can apply. Maybe somebody else knows, Sanjay or,
Are there any noise-related issues in this particular GAL3, GAL80 kind of thing that the experimentalists could let us know, which are possible to read to a modeling of some kind?
I think Bhat can answer.
Yeah, there's a lot of noise here in the GAL system. People have characterized that. Yeah. I'm sorry. Hello? So people have shown noise in GAL system. So you can sort of amplify the noise or reduce noise by changing the Gal-80, Gal-3 concentrations. So the purpose of this autoregulatory loops is to sort of reduce the noise as much as possible, but there's a limit to which you can reduce that. So there are papers on this.
There are papers on this.
Yeah.
Okay. I'd like to have a look. Okay.
So
if you look at the expression level and whatever variation it is, it comes from small numbers of regulatory proteins or from the polymerase itself. It comes from basically these are all discrete numbers of molecules. You might have only a few regulatory proteins available. And then it comes from the translation process, as I said. So those are the sources of the noise. It can come from the environmental factors. But you had something to say about that? Or?
No, yes.
In that Thomas Krieger paper, it's transcription, right? Transcription with the image. Whereas the Elovitz papers, it's the final protein products, CFP and YFP.
You can worry about both of them.
Huh?
Yeah, usually the protein product is what is relevant if you want to worry about the effect of noise.
But ultimately, you see, all of this is transcription, right? So
yes, no, these are not, we moved on from noise here, so this is just regulatory circuits.
But let me just make one point. The origin of noise can be anything. It can be a noise because of transcription. It could be a noise in the rate at which degradation occurs. I mean, when you look at the phenotype, you cannot discriminate between these noises. If you want to measure transcriptional noise, you have to measure only the RNA output. So when you say noise, probably you may want to define what exactly you are measuring at the end of the day.
So that is contingent on the experiments that you do.
Experiment contingent upon the output you are measuring. So what I am trying to say is that whenever people talk about noise, people talk about transcription, degradation, or even partitioning error during cell division. It can cause a lot of noise. So people have not incorporated the noise that gets contributed because of the conformational change of proteins. That itself can add to the noise. Forget about degradation rate, transcription, and all those things. So the actual noise in the cellular system is much more than what we think it is.
But if you plot, say, the CV square against the mean, if you plot the CV square against the mean, for most genes in yeast, you get a line like this. Most genes fall on that line, which is just simply explained by a Poisson noise. But there are specific modules. Stress-responsive genes generally tend to be more noisy than, say, housekeeping genes. Also. But this you can do genome-wide across many genes, in yeast at least.
But there you are measuring transcript, right?
No, you're looking at the protein. Yeah.
So also coming back to the aspect that you're saying, so we have looked at cases of this conformational noise also, where, so because specifically in the context of disordered proteins, where there is no one particular conformation, they can actually go back and forth, and that can lead to Also, in the context we have looked at, this conformational noise can actually also lead to non-genetic heterogeneity because of the way they are connected to.
So, but there you are measuring the phenotypic output?
Yes, finally we are measuring the phenotype.
So you have some mutant proteins?
There is an intrinsically disordered protein which under different levels of phosphorylation can change the circuitry in which it's involved.
Okay.
And that can give rise to non-genetic heterogeneity. In the context of prostate cancer, we have looked at that.
On to, so evolution by gene duplication. So this is a book from 1970 by S.Ohno, who more or less originated this entire hypothesis. There was a few papers before that. But the question is, how do genes evolve? Just given a sequence of DNA and some random mutation model, the chances of something functional evolving from that is pretty tiny. So his hypothesis was most genes evolved from previous genes. And they evolve as a result of duplication events, which could be just segmental duplications inside a chromosome, or it could be anti-chromosomes, or occasionally entire genomes, as in Saccharomyces cerevisiae. And once you have 2 copies of a gene where you used to have 1, then you don't have a fitness impact of mutating one of those copies, because the other one is still there. So that was the basic idea, that most genes evolve from earlier genes. And there's enough evidence for this. If in humans, you have multiple copies of genes that exist in 1 copy in yeast, and you have 4 copies of most hogs genes where there's only 1 in fruit fly, things like that. So what happens when you have 2 copies of a gene? So there are several scenarios. The one from S.Ohno was what's called neofunctionalization now. That is, 1 copy stays as it is, the other is free to evolve and develop new functions. The most common scenario is that 1 copy just gets lost because you don't need 2 copies, and mutations can kill off 1 copy. You could also have cases where both copies are conserved and stay more or less the same, maybe because of dosage effects, increased fitness, or whatever it is. Or this is a scenario that has been pushed more heavily in recent years. The 2 copies evolved to play slightly different functions, which were played both by the original. So an example was the ancestral GAL1 and the descendant GAL1 and GAL3, where I think one of them lost its kinase function, but the other, anyway. So, or you can have some copy of these, some sort of combination of these, which where you start off by neofunctionalizing and then you subfunctionalize or something. Right. So there's a lot of literature on how this works, as well as a lot of papers on examining. So this is individual examples have been seen, and I think there are individual examples for all 4. So so there's a paper by Lynch and Force where they push for subfunctionalization as the dominant thing that happens. Well, the dominant thing is that something gets lost, but next to that. And they calculate the probability that a duplicate gene will be preserved by this method according to some evolutionary model. And they find numbers which are actually kind of similar to, under various assumptions, they get a 20 to 50% probability of keeping a gene under the subfunctionalization approach. And the number in yeast is, I think, like 30%. After the whole genome duplication, 70% of the genes got lost, of the duplicate genes, but 30% survived. Their observation is that subfunctionalization becomes more likely also in smaller effective population sizes. So by which they mean, I think 100,000 is the size of the population. Smaller than that, you have a higher likelihood of subfunctionalization. And in their model, larger populations, you sharply drop off the probability of subfunctionalization. So they call this the duplication degeneration complementation model. So after you duplicate, you're free to degenerate because mutations add up and they don't affect your fitness as much. But because both copies can degenerate at different places, it might end up that you need to retain both.
Rahul?
And then
Sorry, maybe I missed what's the link between this idea and the previous set of ideas?
There's no link. I'm just talking about, these are 4 different things I'm talking about in this. there's the only link is that they all cropped up in my mind when listening to Bhat. So, yeah.
I was wondering if it was.
Yeah. Yeah. Okay, I'll take a look here. Right. So this was an early paper in yeast after all these other genomes were sequenced. Cerevisiae was sequenced much earlier, but in 2004, Bionus and all these other yeasts were also sequenced, and these people looked at what happened to these multiple copies. So basically, there was some doubt at this time about the whole genome duplication, so they claimed they are the first to establish it conclusively. They compared with K. volti, which is a close relative of K. lactis, I think. They basically say that, so they don't go into the sub-functionalization, neo-functionalization thing, but what they say is that in these duplicated pairs, typically one copy evolves much faster than the other, which kind of tends to support the neo-functionalization idea, that one copy retains the ancestral function and the other does something else. So there are many examples of that in yeast. This paper looks at what happens to regulatory networks. So you had one regulatory network, and after duplication, now you have two. And it turns out these networks seem to retain some basic structure and so on. And the last topic, I think we have only like 2 minutes before the scheduled lunch. This is something that's kind of interesting to me, and it came up, as I said, in this case of why do a few cells decide to express this alpha-galactokinase when it is actually of no benefit to them to do so. And there are many other examples in biology of, especially in the animal kingdom, of individuals doing something that they all share the same genome, you remember. So presumably, if they kill themselves for no reason, that's bad for, those genes will be lost. So it's not the survival of, fitness operates mainly at the individual gene level. So there must be something in this altruism genes, as you call it, that helps the fitness of the species. Another example is in ant colonies. The workers and the queens and so on have exactly the same genome. But the workers don't reproduce. They sort of move into a fate that is non-reproductive. And how is that helpful? So it's helpful for the whole colony. But this question has also been studied a lot in the economics literature on why would you... in the real world, people are altruistic, but economists like to believe that people are selfish. And so why would altruism maybe help you? Just by coincidence, I found this article today. It came out yesterday, I think, in Current Biology. So if anyone wants to read it, this is one particular example of altruism, which is skin selection. The idea is that you're more likely to sacrifice yourself to save your close relatives than strangers, and they've observed that in the wild as well. Okay, so this is one of the early papers on how cooperation might have evolved. So how many of you don't know the prisoner's dilemma game? You all know it, or okay, a couple of you don't. So just to iterate, the idea is that you have two prisoners in a jail, and you suspect them of having committed a crime, and they were both in it together, but they are not confessing. So you offer them a deal that if they confess, they get a reward. But that's if they both confess. So that's called cooperation. So let's say cooperation is a confession. So, and if one of them cheats on the other, that's called defection. So they get rewards which are like if they both cooperate, then they both get a reward which is called the sorry? Don't mind. We can do it later. Yeah. Okay. So basically, that's the. It turns out there's something called a Nash equilibrium, which means it's basically the choice of decisions that the players make that if neither of them has any incentive to change unilaterally. So if I choose a strategy and any other strategy is bad for me, then, and if that is true of every player in the game, that's called a Nash equilibrium. And the only Nash equilibrium is if they both defect. That's obviously bad for both of them because if they both cooperated, they'd get better. So to resolve this, people have done things like iterated prisoner's dilemma. Again, it's Axel Roth who did a lot of that work in the 80s. So the idea, he actually simulated this by asking lots of people to play this game, where now you choose a strategy based on the history of the game. And it turned out the strategy that performed best empirically is tit for tat, which is you do what your opponent did last time. And that maximizes your rewards. But anyway, that was more of interest to economics literature. But anyway, so yeah, so there's a, relating to this, I think I forgot to add a couple other references, but there is a, as I said, various mechanisms proposed for altruism, which is reciprocal altruism, which is more relevant to what I said, I think, the prisoner's dilemma and so on. There's inclusive fitness and multi-level selection, which are probably what's happening in the seized case. Why do some individuals in a clonal population act differently in order to benefit the whole population at the expense of benefit to themselves? Right, so these were some of the cases where, theorists might find something interesting to do here, I thought. But of course, at a, more larger level, theorists can basically simulate the, regulatory network at a, people have done that kind of work, including you and KV Venkatesh, right? Simulating regulatory networks in yeast that predict gene out, outputs and so on. So you can have, complicated linear, coupled differential equation systems that do this. So that kind of work has been done for a long time. Anyway, so I think it's basically lunchtime. Do you want to?
Sorry, let's thank Rahul first.